Namaste everyone, I am Dr. Amit. Welcome back to my YouTube channel at the rate Dr. Amit Official. If you are new here, please do subscribe for more simple and high-end medical lecture. So in our previous video, we have already discussed glycolysis and the TCA cycle. So now it's time to move one step ahead, the electron transport chain and oxidative phosphorylation, okay? And we all know that this is one of the most important high-yield topic in MBBS professional exam, USML exam and post-graduation exam. So let's understand this topic in a very simple way and master it step by step. So let's start electron transport chain and oxidative phosphorylation. So let's first understand that this electron transport chain and oxidative phosphorylation, it is a final pathway of cellular respiration where NADH and FADH produces ATP. We all know that in glycolysis we get NADH and in TCA cycle we get NADH and FADH. So that NADH and FADH enters this ETC and oxidative phosphorylation to produce ATP. Okay. So simply what happened that this NADH and FADH transfer electron and creates and pumps the proton and creates a gradient and makes ATP. Okay. So to understand this ETC and oxidative phosphorylation, we must understand this figure. Okay. So in this figure, we can say this area is mitochondrial matrix, and this whole area, this painting area, is inner mitochondrial membrane, and this is in intermembrane space. Okay, so let's understand this figure that you can see this. Okay, so we can call it as a complex 1. This is complex 2. This is complex 3. This is complex 4, and this is complex 5. All these complexes helps NADH and FADH to produce ATP. Okay, now let's see. In between this complex, we can see it is a coenzyme Q and cytochrome C. Okay, so let's understand what is this. So coenzyme Q is a lipid soluble carrier which moves freely in the membrane. Okay, it moves freely in the membrane and it takes electron. Okay, electron from this complex and this complex to complex 3. In the same way, cytochrome C. Okay, this cytochrome C is a small protein between complex 3 and complex 4, and it carries protein from complex 3 to complex 4. Okay, now we'll see that what actually happened in these complexes. Okay, so let's understand this from this paper. Okay, so we all know there's ETC and oxidative phosphorylation occur in inner mitochondrial membrane. Okay, in this place. Okay, now while talking about the function, so it produces ATP using electrons from NADH and FADH2. So the NADH and FADH2 of TCA cycle and glycolysis. Okay, it provides this electron to these complexes. Okay, and this electron will move in this inner mitochondrial membrane and at last it produces ATP. Okay, so it's written here. Okay, so produces ATP using electron from NADH and FADH2, right? Okay, now the basic concept is this NADH and FADH are electron donor. Okay, so we can see in this image that this NADH is giving 2 electrons and this FADH is also giving 2 electrons to these complexes. Okay, now the second thing is electron moves through complexes. We all know that after providing electron to these complexes, this electron start moving in these complexes and at last produces ATP. Now energy used to pump H plus proton. Okay, so we can see here that all the complexes are pumping proton H plus out. Okay, so this is written here that energy used to pump H plus proton. Okay, now it creates a proton gradient. Okay, so when electron enters in the complexes, it pumps out proton. Okay, and it creates a gradient. Okay, difference in charge over here. Okay, now it's written here that NADH electrons are transferred to complex 1, FADH electrons are transferred to complex 2. Okay, so we know that NADH is getting produced in glycolysis and TCA cycle. So NADH give electron to complex 1, but the FADH produced in TCA cycle, okay, it gives electron to complex 2. Okay, always remember this line. Okay, then it's written that the passage of electron result in formation of a proton gradient and coupled to oxidative phosphorylation and drives ATP productions. Now we'll see all the complexes. Now let's talk about complex 1. Okay, so complex 1 is NADH dehydrogenase. Okay, so what's the function of complex 1? Transfer electron from NADH to coenzyme Q. Okay, so this is complex 1. Okay, so NADH is giving its electron to complex 1 and complex 1 is transferring this electron to coenzyme Q. Okay, it's written here. Then pumps 4H plus out. Okay, so while receiving 2 electrons, this complex is pumping 4H plus out. Okay, now the next thing is NADH is NAD plus plus E electron. Okay, so while it forms NAD plus and gives 2 electron, then there are 2 inhibitors. Okay, rotenone and amytal. These two things can inhibit this complex. Okay, this can inhibit the complex and can inhibit this process, providing electron from NADH to complex 1. Okay, now we'll talk about complex 2. Okay, now we'll talk about complex 2. Okay, so complex 2 is succinate dehydrogenase. Okay, its function is take electron from FADH2. So we can see that FADH2 is providing its electron to complex 2 and passes to coenzyme Q, also called as ubiquinone. Okay, so now this complex 2 is giving electron to COQ, and this electron will be transferred to complex 3. Okay, so important point is it does not should pump H plus. Okay, so all the complexes give H plus, but this complex do not give H plus. Okay, so this is a reason why FADH produces less ATP than NADH. Okay, so what are the inhibitor of this complex? So malonate and carboxyl, these two can inhibit this complex, and the process will not go further. Now what's the pathway? We can see that FADH give electron to complex 2, then it is going to COQ, then it's going to complex 3, then cytochrome C, then from complex 3 it's going to complex 4. Okay, and then complex 5. Okay, but when you see in this NADH will give electron to COQ, then complex 3, then cytochrome C, then complex 4, then complex 5. Now we'll talk about complex 3. Okay, this complex. Okay, so it is also called as cytochrome BC1. Okay, so it transfers electron from COQ to cytochrome C. Okay, so the main function of complex 3 is to receive electron from coenzyme Q and to send received electron to cytochrome C. Okay, and it also pumps 4H plus out. Okay, and it is inhibited by antimycin A. Okay, this complex is inhibited by antimycin A. Most of the time in USMLE they ask question from these inhibitors. Okay, now we'll talk about complex 4. Now while talking about complex 4, it also called as cytochrome C oxidase. Okay, its function is to transfer electron to oxygen and forms water. Okay, and pumps 2 H plus out. So you can see that this is complex 4. Okay, this complex 4 actually receive electron from cytochrome C, and after receiving electron it gives it to oxygen. Okay, and it forms water. Okay, I'll tell you this. Okay, now very important is final electron accepted is oxygen. Okay, so what are the things which inhibit the complex 4? That's cyanide, carbon monoxide, and azide. Okay, this can inhibit complex 4. Okay, so these stop the electron transport chain and causes cell death. So what actually happen is this complex 4 give the electron to water. Okay, and it forms H2O. Then you can think that what 2H plus is doing here. So actually if oxygen and 2 electron combine together, then that's a unstable form. Okay, so to become a neutral molecule it attracts 2H plus. Okay, so this electron come from ETC and H plus come from the mitochondrial matrix. Okay, so from here they get this 2H plus. Okay, now I'll talk about complex 5. Okay, this complex. Okay, it's also called a ATP synthase. Okay, actually all the complex is pumping out H plus. Okay, but this complex 5 is actually taking this H plus again back. Okay, to synthesize ATP. Okay, so H plus come back and drive ATP formations and convert ADP plus PI to give ATP. Okay, it's also called oxidative phosphorylation. Okay, and the inhibitor of this step is oligomycin. Okay, so in this step actually ATP formation is occurring. Okay, so this is all about the 5 complexes. Okay, that complex 1 is taking NADH, complex 2 FADH, then they are transferring electron to COQ, then complex 3, complex 4, and complex 5. Okay, now we'll talk about uncoupling protein in the part 2 of this oxidative phosphorylation. Okay, so if you like this video, please do share with your friends, and I'll upload the second part soon of this ETC and oxidative phosphorylation. So please subscribe my YouTube channel, and thank you for watching. Thank you.